This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
So Michael Burke. Michael's the Alfred Deakin Chair of Psychiatry at Deakin University and also leads the Impact Institute, which is a, multidisciplinary institute that looks at innovative treatments in the mental health and sphere and more broadly. So Michael's one of the preeminent mood disorder experts in the world, and also one of the definitely Australia's leading clinical trialists in the mental health space. So I was been a very important mentor to me. I had the great fortune of working with Michael on my first clinical trial, and it was an incredibly valuable apprenticeship. Now leading the Magnet Network, so it's Australia's first clinical trials network for mental health. And Michael has a lot of talents, but also one of your major talents is your ability to think about novel approaches to treating mental illness and repurposing medication from the rest of medicine for mental ill health. And you have extraordinary success in getting grant funding for all these ideas. So I'm going to be very keen to hear what you have to say, Michael.
Well, thank you very much. It's truly a pleasure to come back to the department, and I have to say I'm honoured to still be officially a member of this department. It's an affiliation I have nurtured and I have treasured. So thank you, and it's great to be here amongst friends. So I'm going to talk a little bit about the challenge of therapy development in the mental health space. The great problem we have is there's a drought in novel therapy development in the mental health space. This is a graph from a biotech company, which looks at number of trials in each area. So if you can look at the bottom, schizophrenia, and you can see over the last 2 decades, the pipeline is thinning, not thickening, which is very distressing. Now, each dot is one trial. And so all the orange squares are the same drug. So this is much worse than it actually appears. The drought is really acute. So we've thought about how on earth do you develop novel therapies? So the path that is used in most of the rest of medicine is what I call path one. You have a coherent pathophysiology, you identify a target, and you identify a drug that binds to that target, right? But no examples of that in psychiatry. Number 2 is you aim at a biological system or a network. And the examples would be inflammation, where you can use COX inhibitors or minocycline for neurogenesis, MAC for oxidative stress. And there's some examples of success there, but it's a fairly blunt tool. But the reality is that almost everything we've got in mental health comes through pathway 3, which is just serendipity. Lithium calmed dogs. Anti-tuberculous drugs seem to make people in asylums happy, and then we got the monoaminoxidase inhibitors. That's psychiatry, and everything else is just repurposed from that and refined. But we're thinking that there's a 4th path, and that is repurposing based on reverse engineering. And I will show you in this talk several examples where we've been able to go through reverse engineering and come up with novel therapies. So drug repurposing, we believe, is a really pragmatic way to help revitalize the field after drought. 30% of recent drugs approved by the FDA are repurposed, and there's success in multiple areas, particularly cancer. It's much quicker and cheaper than traditional drug discovery because you have a molecule. And you know these drugs. You only pick them if they're safe and they pass safety testing and they're tolerable. So this is my blue sky vision. It's kind of what we've been fighting to develop over the past 2 decades, really. So we start with discovery models, which can be stem cells, they can be biomarkers like inflammation, oxidative stress. We then validate this through epidemiology and through animal models, and then we can move to clinical trials and on to dissemination. But our goal is to develop new treatments that are safe, that can manage the challenges that are unmet by current therapies. That's our big picture vision. And what I've tried to do in the last 20 years down in the wild, wild west of Geelong is establish a research infrastructure that matches each one of these goals. So what we do is we start with stem cell models, and I'll explain what we're doing, but essentially we're using gene expression as our transcriptomic readout. We do animal models of depression, mania, cognition, pharmacoepidemiology, and onto clinical trials. And we now are fortunate to have Credit, which is our CRE, which funds the discovery platform, and Magnet, which is the clinical trial network with Chris and as our partner. And we're delighted to be working with you on Magnet. And that's how we go on and validate them, because it's a funnel, right? Much easier to identify a compound than to validate it in a trial, and much cheaper. So this is our book, and I'm going to go through 6 chapters quite briefly on the various things that we're working on as pathways to novel therapy defaults. I'm focusing on mood disorders, but we do work in other areas. I don't have time to show you all the trials that we're doing in our group. So we started off with aspirin. And the reason why we started off with aspirin is, again, we started with a biomarker that we targeted. So we know that stress, early life adversity, medical comorbidity all lead to peripheral inflammation, and inflammation, it activates microglia, it changes neurotransmitters like serotonin and glutamate, it leads to oxidative stress, and it results in mood symptoms. So it should be a very straightforward thing: reduce inflammation, treat depression. Easy. So we're interested in aspirin, particularly low-dose aspirin, because it's really tolerable. You can take 100 milligrams of aspirin, it's really tolerable, it's widely used, it's got a good safety profile. And here's I'm just walking you through the journey that we go through, right? So in this case, this is not our animal studies, other people have done this. But in this preclinical model, if you look at the forced swim test, which is the best established model of depression, you can see here that aspirin reduces depression in the same manner as amitriptyline. So chronic mild stress induces immobility, aspirin reverses it. That's the readout you want, and that suggests that it might be an antidepressant. We then go to epidemiology. So what we do in our own Geelong osteoporosis study, because it's a big health cohort where we can look at people, follow them over a decade. And in this study, you can look at people exposed who are taking, in this case, aspirins or statins, people compared to those who are non-exposed. People who take these drugs have a dramatically reduced risk of developing depression over the 10-year follow-up. I don't have time to show you, but I can tell you we've published now 10 papers replicating this finding, showing that if you're in the general population taking aspirin, your risks of depression are lower. There's one clinical trial of aspirin coming out of Iran, positive. So if you take aspirin or placebo, add on to sertraline, you get better. One always has to take these studies with a large grain of salt. There's some methodological and other challenges in interpreting these studies out of Iran. But yeah, it's nice to see. So I had the privilege of being able to partner with John McNeil at Monash on the ASPRI study. ASPRI is a truly awesome clinical trial. So randomizing males and females over 70 to aspirin or placebo, following them for 5 years, 100 milligrams. The sample size is 19,114 participants. So in terms of the depression study, it's the largest clinical trial ever done in psychiatry, as far as I know. And they're annual follow-ups, and we asked 3 questions. Can aspirin reduce the risk for depression? Our primary goal was primary prevention. Because roughly 10% of this cohort had depressive symptoms at study entry, we then could ask, does aspirin treat index depression? And then we wanted to know, do inflammatory biomarkers indicate depression risk, and does that predict response to aspirin? We haven't got the answer to that yet. I have a PhD student onto that. So this is a constant flow chart. 83,000 people were screened, 19,114 randomized to 2 groups. And these are our results. This is the preventive capacity of aspirin. Now, when you see this graph, you can send the statistician home. There's absolutely no signal whatsoever. It's utterly useless. Doesn't do anything to prevent depression. But we still had a glimmer of hope that in the group of people who were depressed, we'd see something. We did, but not what we thought we were going to see. Makes it worse. Aspirin increases depressive symptoms in the 5 years after people who are depressed were randomized to aspirin. Now, this was not exactly what we were expecting to find, but you often learn more from your failures than your successes. That's a really interesting mentorship lesson, but that's a different lecture altogether. But now we start to think why. And so now the next generation of studies, and this is this PhD students after this, we're looking at Is this due to impaired permeability? Is this through changes to the arachidonic system? Why is it making people worse? Because if we can understand why aspirin is making people worse, it might give us an important window into what the pathophysiology of the disorder is. So chapter 2, statins. Statin has many effects that make us think that they could be antidepressants. They have robust effects on cardiovascular risk factors, reducing atherosclerosis, lipids, and glucocorticoids. They enhance neuronal plasticity. They have effects on many neurotransmitters. They reduce oxidative stress, and they're significantly anti-inflammatory. So there are many potential mechanisms of statins suggesting why they might work. Same journey I'm taking you through. If you look at preclinical studies, I'm not going to bore you with the details, but in this chronic mild stress model, simvastatin was as good as imipramine in reversing chronic mild stress-induced depression. So that's always nice to see. In terms of epidemiology, there are now a gazillion studies. And in this meta-analysis, you can see a clear protective effect of statins on depressive symptoms in population studies. Now, my good friend John McGrath has significant skepticism about these pharmacoepidemiology studies because of the robustness of residual confounding in these data sets. So you really do have to interpret them with a large chunk of salt. And in the end, it all boils down to clinical trial data. But I'll show you one of these studies. So this is a Swedish cohort study where they looked at the whole population of Sweden. And you can see that in people who take statins, there's a dramatically reduced risk of depressive disorders. And we've published probably 6 or 8 studies showing exactly the same thing. I'm going to show you one clinical trial just because I like the way it was designed. So it's statins or escitalopram or the combo in people who are depressed after they had coronary syndrome. So just focus on the graph on the left, HAM-D at 24 weeks. What you can see is that the people who got the combo of escitalopram and the statin had the greatest reduction in depressive symptoms. The statin and the SSRI were roughly comparable, and the people who got neither did worse. And this is nice clinical trial data. This gives you a bit of confidence. There is data in schizophrenia, and interestingly, there's a reduction in negative symptoms in people with schizophrenia with statins. So these are now half a dozen clinical trials that have been done. Nothing much for positive symptoms, but negative symptoms, it is statistically significant. And Bob and I were talking about this earlier. Really good data on statins and cognitive decline. So this is a really nice study which looked at fairly mild cognitive impairment. So the top cognitively normal people where it can't change cognitively normal people. And if you've got in the bottom to late mild cognitive impairment or frank Alzheimer's disease, the horse is bolted already. But in early mild cognitive impairment, there's protective effect of statins. So statins seem to have non-specific neuroprotective effects across a diversity of syndromes. So Chris and I, when we were doing this study together, when we were both working at Origin, we partnered in the YODA-A study. So YODA-A stands for the Youth Depression Alleviation, and A is anti-inflammatory. It's a 3-arm study, aspirin, rosuvastatin, placebo. It's a 3-month study with a follow-up at 6 months. And this is what we found. At baseline, the groups were nicely matched. At endpoint, the difference between rosuvastatin and placebo just missed statistical significance. We had the great problem at the time of running 3 depression studies simultaneously. And so all studies struggled to recruit because you can't do 3 studies in the same place at the same time. So we missed our sample size. So I think this is just a power issue. Had we met our sample size, I think this would have been a positive trial. But aspirin was statistically inferior to rosuvastatin. So the statin beat aspirin, and that was statistically significant, but we didn't quite get significance on the primary endpoint, which is a terrible shame, because then you have to report this as a negative study. But there's now enough trials that you can look at meta-analyses. So this breaks it up into baseline 2 for 8 and 12 weeks. And what you can see here, by 8 weeks and certainly by 12 weeks, statins are statistically significant. And I think this is particularly important for youth depression, where SSRIs probably are not that effective. And if statins are effective for youth depression, and they're really well-tolerated agents, they're much more tolerable than antidepressants, I think that could be quite useful. But we died a death because we weren't powered to claim a statistically significant result. On acceptability, which was what I was hinting at, it doesn't separate from placebo. The diamond is exactly on the top of the line, showing a very good safety profile. There's more recent data about which is the best statin to use. So this network analysis was published in October of last year, and it suggests atorvastatin may have the edge in terms of antidepressant effects. So again, there are a couple of studies going on now that I want to just tell you about. One of them is STARI. So this is led by Sophia Zungas at Monash. So this is very similar in design to ASPRI, elderly people over the age of 70, 5-year study, and it's atorvastatin or placebo. So the aim is to look at whether statins reduce the incidence of major depression. In other words, it's prevention, and then there's also a therapeutic goal to see if you're depressed at study entry, do statins treat this? So given everything else I've shown you, I'm more hopeful that STARI will be positive than ASPRI, but you got to have two-tailed hypotheses in this business because you're always surprised. So let me just skip to diet and nutraceuticals. Food and mental health was not a topic 10 years ago. Nobody paid it any attention. It didn't exist as an area of interest. But there have been absolute dramatic changes in the consumption of food over the past 2 decades. At the moment, if you look at the US data, US kids get 60% of their calories from ultra-processed junk food. This is an unheralded change. And this has massive public health implications, including, I would argue, for mental health. If you look across the epidemiology data, and Felice Jacker in my group did the first study looking at this, so she's really the great godparent of the whole field. There's now meta-analytic data that if you consume a Western pattern diet, if you look at the bottom, your odds of developing depression are increased. If you have a healthy diet pattern, it's decreased. So again, huge risk of confounding by indication. So trials are always necessary. So Felice did this study called the SMILES trial, comparing a Mediterranean diet with a psychotherapy intervention. So this was a social support psychotherapy control group, so an active control, not TAU, which inflates effect sizes, against diet. And what she found was, if you compare the diet intervention to the psychological support condition, a far greater reduction in depressive symptoms in the diet intervention group. If you looked at the moderating effect of adherence, so here you can see you break adherence down into 4 quartiles. Quartiles 1 and 2, you didn't really adhere to the diet, your depression didn't change. In quartile 4, you adhered to the diet and your depression symptoms went right down. This has now been replicated several times. I'll just show you a couple of studies. This is Natalie Pauletta's study from Adelaide, showing almost identical results. Again, comparing Mediterranean diet to a psychological support group condition. And another paper published last year comparing dietary change to a habitual diet control group, showing again statistical significance. And this is the 4th study that was just published a few months ago. They called it, this is not our study, but it was, you can see in blue, the Mediterranean diet, a significant reduction in the 5 well-being index. So there's now 4 RCTs, all showing the same. And on meta-analyses, you can see that there's a clear, this slide shows you population-based studies, not clinical trials. I've showed you clinical trials. This is population-based studies. And you can see even in population-based studies, you can see a significant positive benefit of diet in the improvement of depressive symptoms. The problem is most people don't want to go on diets. Most people want to continue to eat what they eat. And they ask the question, doc, can I take a pill for that? Surely I could just go to the vitamin store and take the missing nutrients, and this is going to fix me. Not an illogical question. So fish oil has been the go-to study, and this was the third in the triumvirate of studies that Chris and I worked on together. So this is a study led by Paul Amminger. And again, it's one of those results where you do not need a statistician to tell you that there was no effect of fish oil. There are a number of pre-existing studies that showed a hint, but in my view, the way that Paul did the study makes the most rigorous study methodologically. I really trust the way he did the study, and I trust this is a true null effect. Some of you might be aware of other big studies published recently. There's a study out of Harvard called the Vital Study. So they used vitamin D and fish oil, a factorial design, utterly useless, no change whatsoever on depressive symptoms. Then Jerome Saris a couple of years ago did the study where he looked at a combo, right? So you looked at all the nutraceuticals for which there's a possible benefit in mood. And so he took fish oil, zinc, folinic acid, and S-adenosylmethionine. So each one of those on their own has got suggestive evidence that it might have antidepressant effects. And what Jerome found was not only was this not statistically significant, but numerically in favor of placebo. In other words, the vitamins did worse than placebo numerically. Now that might just be by chance, except, come on. This has now been replicated. So this study, which was published last year, looks at a different combination. This is fish oil, magnesium, folic acid, and B12. And numerically, the placebo group is doing better. The vitamin group is doing worse. Now, think about it. 70% of the Australian population is taking vitamins. Most of them are taking them because they just want to feel better. In other words, for mood, quality of life, and it's making people worse. So this message isn't out there. So one of the studies that we're doing, this is led by Adrian O'Neill, and it's part of the Magnet group. It's called Hearts and Minds. Well, actually, the name's been changed to Harmony. So the aim is to combine diet, exercise, smoking cessation, management of substance abuse, and using this study in a group of people with depression. So the hypothesis is that the lifestyle program will be non-inferior to gold standard formal CBT. It's a very ambitious study, aiming to get 600 people. She's just finished a study using the same intervention as a pilot, was able to rapidly randomize 180 people, and it was non-inferior to psychotherapy in the pilot. So that's really encouraging. So if lifestyle can genuinely be non-inferior to standard CBT, with all the attendant other health benefits in terms of dementia and cardiovascular disease, that's kind of clinically interesting. You want to get crazy? Fecal microbial transplantation. There's a couple of case reports of FMT for the treatment of psychiatric disorders. This is a case report published by Gordon Parker, showing a significant reduction in depressive symptoms in a patient with bipolar disorder. And we have just finished this study called the Moving Mood Study. It's a pilot, not powered for efficacy. It's a feasibility study called the Moving Mood Study. So we're using FMT as an adjunctive treatment for major depression. And we hope to have, the paper, I've just seen the final version of the paper, so hopefully it'll be out soon. All right, so if that's not crazy enough for you, let's just rank, ratchet up the craziness a little bit more. A paper was published in the Deacon group a couple of years ago, where they used genetic data. In this case, they used SNPs from the Wellcome Trust data set. And what they did is in the Wellcome is they had SNPs for 7 complex diseases, and then they could look at a gene triplet analysis, which said, what drugs do these SNPs target? So what drugs might be effective at this genetic signature? So these, remember, this is SNPs, not gene expression, not transcriptomics. But it comes up with a set of potential drug candidates, and it comes up with 2 drugs. Imipramine, which is pretty nice if you're interested in an antidepressant. I would think of that as a positive control. And it comes up with an angiotensin drug, valsartan. Now, this was published, 8 years ago. We didn't know what to make of this, and we put it in the filing cabinet. Then I'll be working with Ken Waldo, who's really my partner in impact. So he's a diabetes researcher. And so what he did, I listened to him give this lecture on this slide. And when I heard him give this lecture, however many years ago, I said, this is the best thing I have heard in the last 10 years. We have to work together. And that was the start of our institute, me listening to him talk to this slide. So what he did is he established a gene expression signature of type 1 diabetes. So you look in rats, or in this case, in tissue culture, at what is the net gene expression signature, the transcriptomic profile of known diabetic drugs. And there's a whole lot of diabetic drugs. And then you look at what other drugs do what these drugs do in terms of gene expression, and he comes up with a glaucoma drug called methazolamide. He then goes to animal models, and in animals it lowers sugar. And then he went to a human clinical trial and showed that it treated diabetes with lowered sugar. And I thought, this is brilliant, because diabetes genetically is not too dissimilar to mental health disorders. It's a complex, polygenic, environmentally mediated disorder. No single gene, lots of genes, all of small effect, environmentally mediated. So I thought, if it works for diabetes, we have to work together. And the rest of the talk is going to be me listening, the outcome of me listening to Ken. So what we're doing now is we're growing stem cells. So we take people with bipolar disorder, we take blood, we reprogram them into pluripotent stem cells, and we differentiate these into cortical networks, and they grow them in test tubes. The choice of cell model is vital for success in GS drug screening. We started off looking at off-the-shelf neuronal NT2 cells, but we're now using patient-derived stem cells, and we're even going further. We're dividing our patients into lithium responsive and lithium resistant. So what we did in our first studies, we used targeted bipolar disorder, and we chose 4 mechanistically completely different drugs: lithium valproate, lamotrigine, quetiapine, because we don't want to just recapture what one drug does. We want to capture the net effect of mechanistically diverse drugs that all work for the same disease. We look at gene expression, we do Bayesian analysis, we come up with the gene expression signature. We then take white cells from people with bipolar disorder and look at that gene expression signature. We look at rats given the same cocktail and look at that gene expression signature. Then we come up with our final choice of genes. We get about a half a dozen genes. We do drug screen, and then we come up with what drugs in screening come up with the same signature as our known drugs. And so for our first one, what do we find? So I'll start you at the bottom. Cinnarizine, a calcium channel blocker. Well, I love that when I saw it, because I did my PhD in intracellular calcium. To come up with a calcium drug, whoopie-doo. Flupentixol, I love that even more, because that's an effective anti-manic drug. So another positive control. Then we come up with an ACE inhibitor again. Our second theoretical drug screen, using a genetic readout, comes up with an angiotensin drug. Ain't that interesting? Then we get a COX inhibitor, and I've already spoken briefly about inflammation, and I haven't shown you the COX literature because we're not doing that work. And then a whole lot of cancer drugs, we don't know what they do. At the same time, another group called Fabry did a study. So they did this after we've already begun our clinical trials, but it was really cool because they looked at treatment-resistant depression And they looked at genes in pathways associated with TRD, and they looked at drug candidates that targeted those genes. And what did they come up with? Telmisartan. So 3 atheoretical drug screening paradigms, all completely different, are coming up with the same angiotensin target. There's also a number of other drugs that we're interested in. So cannabidiol, well, people are studying that. They're coming up with dopaminergic agents, pramipexole, which we know works for depression. So ketamine, well, I don't have to tell you, ketamine is flavor of the month for depression. So this atheoretical screen comes up with 4 drugs that are already in current clinical development for depression. So gives us confidence that it's finding something. The next step is you go to animal models, and I'm not going to talk you through the data, but I'll just tell you, in animal models, candesartan has antimanic effects. Losartan, which is first cousin of candesartan or sibling, it's got antidepressant effects. In this case, this is a sucrose preference test, which is the other screening test for depression. And you can see if you trigger depression by giving lipopolysaccharide, you can rescue this by giving losartan. Candesartan is angiotensin drugs are really good for pharmacoepidemiology because you can control for all the things that normally you can't control for in pharmacoepidemiology studies. So I'll show you this one because it's one of the earliest. So this looks at a Norwegian study called the Hunt study, or the Hunt study as the Norwegians pronounce it. And you can compare beta blockers, which slow a non-significant but slight increase in depression risk, calcium channel blockers, which do nothing, and ACE inhibitors, which halve your risk of developing depression. So I like this. I think that you can trust this because there is an inbuilt control, because the choice between antihypertensive drugs is pretty line ball. When we went back to our epidemiology cohort, this is the Geelong study, we got this Kaplan-Meier curve, and I have never, ever seen one that looks like this, ever. So if you're taking an angiotensin drug in the red, you just don't get depressed. It's almost unbelievable. It's such a robust result. And that the blue line, of course, is if you're not taking the drug. And that's a 5-year follow-up. I mean, truly striking. Now, we have replicated this in multiple cohorts, mainly in Danish and Swedish national cohort studies. I'm not going to show you all that data, but just take it as read. We've done it. But the other thing about these drugs is that the cardiology literature uses quality of life in many of their measures. And if you look at the factor structure of quality of life, it doesn't look terribly different from the depression rating scale. And here you can see significant benefit in cardiovascular prevention studies for these drugs improving quality of life. And this allowed us to go on and do the cadet studies. So we've got 2 studies, the cadet, which is the candesartan adjunctive bipolar depression trial, and the candesartan unipolar depression trial. And these are currently running at 6 sites. So we're running at Melbourne Clinic, and Marta is kindly working on that one. Melbourne Clinic, we're running in Queensland, and we're running in Geelong. So, and also in Eastern Health. So we have 3 Melbourne sites for the cadet trials. All right, if that's not crazy enough, let's ramp it up again. Metformin. Start again. Yet another atheoretical drug screen, and metformin comes up as a candidate that we identified as having potential efficacy. So this is, again, this stem cell discovery model. We identify metformin. There's quite a lot of preclinical literature. So this is a study that looked at both antidepressant and anti-anxiety and antidepressant and antipsychotic models. So the antipsychotic model is pre-pulse inhibition, which is a pretty crappy model, but it's the best we got in psychosis. It's positive. So metformin is protective. And in both the forced swim test and the sucrose preference test, which are the best animal models of depression, metformin is protective. There's a really nice study coming out of Taiwan by Mark Valquist. So this is a whole population of Taiwan. And you just focus on the top left-hand corner. So it's probably a little bit hard to see, but you can see here, if you look at all affective disorders, if you have no diabetes, your odds ratio is 1. It goes up to about 2.6 if you have diabetes, and back down to 1 if you take metformin. So that's nice data. So in this data set anyway, metformin appears to be protective against depression. This is our epidemiology data. So women taking or not taking metformin. And you can see a clear protective effect of metformin. Remember that you take metformin because you're fat, you're insulin resistant, you eat junk food, you don't exercise. And those are all risk factors for depression. So if this was confounding by indication, you'd expect the metformin group to do worse, not better. The fact that they're doing better, it's kind of interesting. There's one trial, comes from Iran. Don't trust it, it's positive. But there's a slightly better trial that was done by Cynthia Kalkin. So she's from Canada, and she published this in the JCLIN site a couple of months ago. It's a pilot, study small. And what she did is she gave people with bipolar disorder and insulin resistance metformin or placebo. And what she showed is that if you reversed the insulin resistance, in other words, you became a converter in her language, there was a dramatic reduction in depressive symptoms in the study. And that's nice to see. So it's an interesting, novel study. Pilot, samples are small, 20 in one group, 25 in the other, but interesting. So that takes us to the next trial that we are beginning, which we're calling Hearts and Minds. So this is a 3-arm study, statins, metformin, and placebo in the treatment of depression. This has just been submitted to Melbourne Ethics, and let's see what they make of it. And I'll finish off by talking about mitochondria, which is probably the craziest of all the things I'm talking about in the lab. It's my view that bipolar disorder is essentially a mitochondrial disorder. If you look at the phenomenology of bipolar disorder is characterized by 2 phases: excessive mitochondrial energy generation and inadequate mitochondrial energy generation, manifested by increased energy and agitation and decreased energy and psychomotor retardation. That's the essence of what bipolar disorder is. So we've published a theoretical model of the biological basis of bipolar disorder. And our argument is that all the biological and environmental factors that drive bipolar disorder converge on mitochondria. Some are stimulatory, like dopamine, glutamate. Some are inhibitory, like uric acid, calcium, BCL2. But they all converge on mitochondrial dysfunction. So that our key hypothesis is that bipolar disorder is a state-dependent dysfunction in the regulation of mitochondrial energy generation. In the depressive phase, you do not generate enough energy. In the manic phase, you are generating more energy. You're unable to stop generating energy when demand goes away. In the depressive phase, you're unable to generate energy in the face of demand. It's a mitochondrial failure, but it's a state-dependent regulatory failure. References below in case anyone has insomnia. The good thing about mitochondria is that it's potentially druggable. So there are many compounds that have some effect on mitochondrial functioning. This figure shows some of them, and some of these are practically druggable. So we've been interested in some of these on the bottom: coenzyme Q10, alpha-lipoic acid, N-acetylcysteine. These are compounds that are safe, tolerable, effective, and might have an impact. So we did a study of mitochondrial cocktail for the treatment of bipolar depression. 3-arm study, NAC alone, placebo, and this combination of nutraceutical factors for the treatment of bipolar disorder, depressive phase of bipolar disorder. So there's now a meta-analysis of these various elements from other studies that have been done, including the study as well. And you can see here that the best evidence here is for NAC. Some of the other ones like vitamin D actually make it worse. But the only one that is statistically significant in meta-analysis is NAC. And this is a network analysis, which again looks at the same thing. Of all the various mitochondrially active agents that have an effect on the disorder, the one with the strongest evidence is NAC. Sorry, I'm missing the slide here. I didn't show you the results, but I have to just talk to you about the results. So what we found in that study, at the end of the 3-month period, there was no difference between any of these groups and placebo. But interestingly, 1 month post-discontinuation, mitochondrial cocktail group was significantly better than placebo. Now, that's hard to explain because it's 1 month afterwards, but something happened such that the mitochondrial cocktail group was doing better 1 month after discontinuation. Negative at the primary endpoint, positive at a secondary endpoint. Hard to interpret. What we also saw was a subtle non-significant increase in manic symptoms with the mitochondrial cocktail, which in a way is not what you want, but it does suggest that you can drive the features of the disorder that are associated with energy with this cocktail. There are other things that drive mitochondrial energy generation, exercise being one of them. And we think that one of the effects, one of the pathways whereby exercise is good for mood is through up-regulating mitochondrial biogenesis in the body. What we did see also is that in our study, there was an interaction between physical activity and treatment condition. So in those people who had high levels of physical activity, they derived benefit from the mitochondrial cocktail. So you might have to do more than one thing. In this case, physical exercise plus the cocktail. The people who were physically inactive didn't get any benefit at all from the cocktail. Very post hoc, you have to take it with a pinch of salt. I'm really sorry, this slide is out of order and I didn't pick that up. No, sorry, it's not out of order. This is in order. Forgive me, forgive me. Go back to our drug screen, right? So we've also, in our new generation of drug screens, so this is again data from stem cell discovery. What we did is we came up with a number of hits for bipolar disorder. And the most interesting one is this compound here called trimetazidine. So trimetazidine, I don't know if any of you have heard of this drug. Well, it's a cardiology drug. It's used for heart failure. It's used for angina. And its net effect is mitochondrial. It's a mitochondrial drug. It enhances the efficiency of mitochondrial energy generation. It inhibits beta oxidation and drives aerobic energy generation. One of the things that happens, again, I haven't got the time to explain this all to you, but one of the abnormalities in bipolar disorders is a shift from aerobic to glycolytic energy generation, which is inefficient and associated with increased pH and lactic acid. So you get subtle lactic acidosis in the disorder. And the thought is that this might help. So trimetazidine has a whole lot of effects. So it reduces apoptosis, it influences autophagy, and through this modification of energy generation, it enhances aerobic energy generation, inhibiting fatty acid synthesis, it drives more effective energy generation of glucose through glucose. We've just completed some preclinical data. So this is data done by G. Kim, who joined our group. This is work conducted at the Florey. And she was able to show that trimetazidine increases oxygen consumption in neuronal cells. And that's consistent with what we saw in cardiomyocytes. We also, this is work that we've done with the pharmacology group back in South Africa, colleagues of ours. So they use a Flinders-sensitive rat line, which is another animal model of depression. And what we were able to show was increased time spent in the open arms of the elevated plus maze, which is a model of anxiety, as well as an improvement in sucrose preference, which is a depressive model, as well as an increase in hippocampal oxygen consumption, which again suggests that this is the mechanism whereby it works. So it increases mitochondrial efficiency. We also saw no effect on body weight or general locomotor activity, but we saw an improvement in the forced swim test with trimetazidine, which is again consistent with a general antidepressant effect. The good thing about trimetazidine is its safety is fantastic. So this is a study of 2,000 people published in the Lancet. And they looked at adverse events, and no adverse events were more frequent in the trimetazidine group than the placebo group, which is not, these are not data you see in psychiatry clinical trials. So this gives us hope that in terms of tolerability, we may be onto something if we get efficacy working. So we've just got funding from the Stanley Institute to commence a trial called the TIDE trial. So this is trimetazidine for the treatment of bipolar depression. And we're just waiting to hear from ethics. And we're hoping to start the study probably in 6-8 months' time. So let me just summarize by saying stem cell models show promise for candidate therapy detection, that inflammation, neurogenesis, mitochondrial function, oxidative stress, and the stress response systems provide novel targets which are not being looked at in psychiatry. Statins, minocycline, I haven't had time to show you the minocycline data, but this is Olivia's work, it's positive. Celecoxib, N-acetylcysteine, metformin, angiotensin antagonists, all promising drug candidates. We think that diet and exercise are really important lifestyle approaches. And we believe that repurposing is the highway to bypass the drought in drug discovery. Let me finish off by thanking my team. I'm fortunate to have an amazing team of people who did all the work that you have seen. And I'm immensely grateful to everything they do. So thank you very much.
Thanks, Michael. That was an amazing journey through your very innovative and left field approach to discovering new treatments for mental illness. The people have questions here, and let's see if there's some online questions as well. Well, I'll ask a question while while other people think of theirs. Michael, I mean, it's very striking just how many of the treatments you're looking at are about treatments for the so-called, lifestyle disorders, for, obesity, high blood pressure, high cholesterol, etc. Do you think these treatments are operating by addressing those factors? Like, is it, do the fact that statins have effect on depression have anything to do with reducing cholesterol, or metformin does have anything to do with glucose tolerance that's affecting depression, or is it a different sort of mechanism? Or...
Well, I think one of the things about depression is that I don't think anybody believes anymore that there's one neurotransmitter that's affecting this. This is a complex function subsumed by a vast array of neurobiological processes that all interact with each other. What's true is that the risk factors for depression at an environmental level are almost identical to the risk factors for cardiovascular disease and depression. So whether it's smoking, social isolation, stress, poor diet, physical inactivity, almost everything that's a risk factor for these so-called medical disorders overlaps almost entirely with the risk factors for these so-called psychiatric disorders. And it's artificial to separate them out because they're just, I think it's differential system expression of overlapping environmental risk factors and overlapping biological pathways. So the biological pathways are all the ones I've spoken to, which drive diabetes and cardiovascular disease and stroke and probably dementia as well. I haven't spoken of dementia, but a huge overlap there as well. So if you think about it from that kind of multi-systems approach, it's not illogical that drugs that target the pathways identified for cardiovascular disease as that overlap with the same targets that operate for mood disorders might have efficacy.
So do you imagine that they might work, better in people who already have some obesity or high cholesterol or whatever, as well as in, say, a young, healthy person who has no indication?
We
of any of
don't
these?
know, right? So the Calkin study took the gamble that it did, right? That she only looked at obese people with insulin resistance. Hearts and Minds said no, we're not going to do that. Our gamble is that it might have efficacy in people who are not necessarily dyslipidemic or not necessarily diabetic, because my punt, and again, in this business, you just take punts. None of us know until the study's out, that it's not the cholesterol or the sugar that is your problem. It's the pathways that in some drive both. So you may get benefits in non-diabetic people with metformin and in non-dyslipidemic people from statins, because we're not interested in the effects on HMG-A. Because we don't think that it's a cholesterol problem necessarily. Although even that's a complicated statement, that's probably inaccurate, because when we look at, sorry, this is so much information. When you look at, we've done pathway analysis of the bipolar meds, and we've looked at all the different pathways that bipolar meds act on. The pathway that is most impacted by bipolar meds is cholesterol biosynthesis, which is really interesting. Every single enzyme in that pathway is impacted by standard bipolar meds to a major extent, more than almost any other pathway.
There's a lot of genetic overlap, isn't there?
Huge.
Bipolar disorder and cardiovascular disease.
Chris.
Michael, thanks. The genetic approach is certainly a nice way to go. And I recall that by one of the studies we did identified the angiotensin system in schizophrenia as well as the ubiquitin system, and it's led us in various directions. I'm wondering, with all of the work that you've done now with many different compounds that you've been looking at, and I know you work clinically as well, how has that affected your clinical practice?
That's a very wise question. Now, look, none of us can, at this point in time, start prescribing metformin and statins and all these drugs and angiotensin drugs for people who just randomly. How it's changed my own practice is my threshold for suggesting to a patient you really should start a statin is if you have any hint of dyslipidemia, well, go for it. Because we know that depression and schizophrenia is as potent as smoking as a risk factor for cardiovascular disease. Now, cardiologists don't normally incorporate that into their Framingham risk matrix. But the truth is the presence of schizophrenia is worse than smoking. Sorry, the presence of it's worse than smoking in terms of your risk factor for cardiovascular disease. So at the merest hint of dyslipidemia, I tell my patients to go into a statin. If you've got even subtle dysglycemia, I suggest you go into metformin. If you're on an antihypertensive, I suggest you're probably better off taking an angiotensin drug than a beta blocker. But that's as far as we can push it now. I don't think we have the data to go further yet. Sorry, Bob.
No, I wanted to ask a question, Michael. It seems to me that there are great advantages in lumping together large groups of people and saying depression or everyone who takes statin and so on. But I think there are risks as well, aren't there? I mean, can you edit this so as to look at subgroups at particular types, say bipolar people with a family history?
Oh, absolutely. Yeah, absolutely. The problem is
if you did that, if it got better, wouldn't it tell you something? And if it didn't get better, wouldn't it tell you something?
Absolutely. Subtyping is one of those areas that everybody's interested in. It's of huge promise, but I don't know that anybody's ever succeeded in defining valid subtypes of any of these disorders. Depression, I know people like Gordon Parker spent his career trying to identify a melancholic subtype, but the evidence really hasn't stacked up. And whenever we go back post hoc and try and stratify for any of the studies we've ever done, have we ever found anything? No.
Suppose you start with one of the genetic conditions, Wilson's disease, something like that, where you have psychiatric sequelae from it. If you started with the genetic disease, would you perhaps be in a better position?
You might. Right? So there's a study like that going on with N-acetylcysteine, and that's Huntington's. So Clement Loy is leading a study of NAC for Huntington's, and then you have a genetically homogeneous condition, and it might be that that's the place to go. So that your approach is currently being looked at.
And in cognition, you could use Alzheimer's disease to the genetic cases of Alzheimer's.
The ApoE, yeah. So you could, absolutely. Yeah.
Thanks, Michael. I just had a question about the specificity of the effects. You said specifically the statins, obviously in the MTI literature, seem to have like a broad neuroprotective effect. But in one of the meta-analyses, it probably showed effects of depression, not anxiety disorders. Are there any thoughts about why that might be the case?
Yeah. Pretty much all the biomarkers that we talk about in depression are not as robustly evident as in anxiety as depression. So inflammation, the association with inflammation, oxidative stress, all these dysregulated biomarkers is much weaker in anxiety than it is in depression. So for that reason, you would be less likely to see an effect of a drug that's modifying those pathways in anxiety than depression. Anxiety is, for most people, much more of a temperamental trait thing, whereas depression is a state thing. It's subtly different. Not always, but.
There's one question online as well.
We can see it here.
Yeah.
So from Beck Cooper, is there evidence for other lifestyle approaches such as sleep as a treatment or prevention for mental disorders?
Well, sleep is huge. There's huge interest in sleep regulation. Dysregulation of circadian rhythms as a risk factor for depression. And there's a huge field on chronobiology of people who are looking at sleep modifying interventions to improve risk for and management of depression. So the Harmony study that I'm telling you about has a circadian regulation module where we help people to regulate their social rhythms as part of the management of risk factors for depression. And then there are weird and wonderful ones like blue blocker glasses. You've heard of that one? Yeah, so you've heard of that. So there's a lot of interest and energy in this. I mean, Ian Hickey, for example, it's his major area of interest is circadian dysregulation. So it's a big area of study.
Well, great. So you've got one more question?
Oh,
Go on,
Tamsin.
Greg. And there's another question online.
I'll take that one.
Okay. Well,
So Tamsin asks, Michael, great talk. I'm wondering if you are collecting cognitive data in any of the trials you mentioned today.
The answer is yes. So in all the Magna trials, we are looking at cognitive data. I have a little bit of PTSD from the process because you should never ask two neuropsychologists for advice about neurocognitive testing. War's going to break out. So we have, and it was a complex process to come up with a battery, but we are measuring cognition.
last question, Ben.
No, I was just curious. Obviously, drug discovery, R&D in psychiatry, it's big pharma has dried up. We all know that. But with some of these other compounds and targets that you're finding, how active is R&D?
Well, the beauty is that you don't need R&D, right? So we don't need to do phase 1 studies. We don't need to do any of the safety stuff that's out there on the market. Some would see it as a disadvantage that there's no IP. Right? You can't take out a patent position on something that's on what, but I see that as an advantage in terms of translation. If you had a business mind, which I don't, you might be upset. But if you have a public health mind, which I do, I think that's not a bad thing.
Hey, Shendo, did you have a last question?
Sure. This process that you've outlined seems efficacious and cost effective. Are other specialties adopting this? So are cardiologists looking into studies on?
I think we are behind. We have much to learn from other specialties. They're way ahead of us, not behind us. So cancer, cardiovascular, these fields are much more sophisticated in their use of these techniques than we are in mental health. And I think we have lots to learn from people, colleagues in other disciplines. They have met many of these challenges already, and we can learn a lot from what they're doing.
Great. Well, thanks again, Michael. Fantastic talk. It's really great stuff. Thanks. Good. Sandwiches, everyone, and